Okay.
Good morning, everybody. Thank you all for joining. It's great having you here in this fantastic webinar that will be held by our guest, Dr. Valerie Budmeyer, Technical Application Manager from Immudex. A new way of exploring immunity with the Dextramer technology, which is already well known. Valerie worked 5 years at the Danish Cancer Society, where the development her expertise for cancer biology, in particular melanoma, using transgenic mouse models, in vitro assays, and bioinformatic tools. She received her PhD in 2019 from the University of Copenhagen and joined Immudex in 2020. So I will leave it here for you, Valerie. Thank you for, it's great having you here.
Thank you for introducing me. I'm very glad to be here, and I hope I can, deliver a good presentation and convince you that this Dextramer technology is a great tool for immunologists these days. Let me just share my screen. All right. Can you see my screen for now? I will put it in full mode.
Not yet. You are the host and the panelist, so you can share your screen. Okay, it's on the way.
How about now?
Yes, perfect.
Yes, and you see it in presentation mode?
Yes, perfect.
Great. Thank you. I'm just going to put this up. Great. So thank you so much. First of all, I want to thank Almog for organizing this. So Almog is our official distributors for Israel, and we're very reliant on distributors like yourselves to be able to reach out to customers. So thank you very much for organizing this. So let me start with giving you a broad introduction to sort of give you some context about at which stage Dextramer technology comes in. So as you know, the immune response is triggered by a series of antigens that can be from bacteria, virus, dust mites, so something external, but also come from within when we developed autoantigens or neoantigens that are produced by tumors. So no matter where this antigen triggered response comes from, it triggers a series of cellular responses within us, either an innate immune response, so something that is encoded in our DNA from birth, or as we grow up, we also adapt to the environment around us and mount what we call an adaptive immune response. And this is in the adaptive immune response are the cells that we're interested in today, which are the antigen-specific T cells. So why are we interested in these highly specialized cells? So as you know, we as individuals will react very differently to the triggers around us, and personalized medicine has become a crucial type of therapy nowadays, because one treatment might not work for all individuals, so we have to tailor these responses to the way our immune system will respond. And of course, in that context, and especially in the context of cancer immunology, understanding which pathway to target and which cells to target has also become very crucial. And in that, the antigen-specific T cells have become a very interesting target as well. And of course, if one can understand which antigen triggers a response, we can use that to develop new vaccines. And once again, for autoimmune diseases, in that case, we want to reduce the type of response that we would get and quiet down the immune system. So understanding which antigen-specific T cells might be triggered in an autoimmune disease will also help us to manage these types of diseases. So a lot of applications and these cells are really involved in a lot of different types of disease. So it's quite important to look at them. So we'll go through some of the key elements that will determine how to use these reagents. And I will start by just mentioning how an antigen is processed within the cell. So it's broken down by the proteasome into smaller bits, refolded into MHC molecule complexes that will be unique for each individual. And this complex is then externalized and presented at the surface of the antigen-presenting cell. So typically dendritic cells, but also a lot of other immune cells have the ability to present an antigen. And so this is what we call the MHC peptide complexes. So we have two different types of molecules or MHC peptide complexes that we're going to look at today. MHC class 1 molecules will host peptides presented to CD8 positive T cells, so cytotoxic T cells, and class 2 molecules will have peptides that will be presented to CD4 T cells. So these molecules change from different species. So humans and mice, for example, don't have the same MHC molecules. And this will be determined by the patient samples or the types of samples you look at. One particularity of the class 1 molecule, you can see they have similar structures, but the peptide binding pocket here is shorter for the class 1 molecule, so it can only accommodate peptides from 8 to 10 amino acids, while typically class 2 molecules will be able to present longer peptides. So I looked a bit at what alleles were present in Israel, in the population in Israel. And of course, that very much depends on also the sort of the genetic background of the patient samples you look at. But one of the most common alleles in Israel is AO2, AO2-1 in particular, AO1, A24, AO3. And for HLA-B alleles, we have B35, B38, B44, and B18. Obviously, if you work with smaller cohorts, these numbers or these alleles might not be in your cohort. So it's always a matter of statistics. And these are obviously very dependent on genetic variability in a given region, or again, bias introduced by a certain disease type. But at Immudex, we cover quite a lot of alleles. So you can see our full MHC allele list here. And you'll see we'll have quite an extensive list of class 1 alleles. We have a few for mouse and non-human primates as well, and obviously some class 2 molecules. So going back to how an immune response is triggered. So as we mentioned, an antigen-presenting cell will expose MHC peptide complexes on its surface. And this MHC complex will be specifically recognized by a single TCR or a single type of TCR on a given T cell. And so what most immunological assay will measure to determine and assess an immune response, we'll be looking at more downstream signaling. So that's cytokine release by ELISA. That's also measuring infected cell death. But this is all an indirect measurement. So if we want to measure active immune response and a direct immune response, we have to look at this interaction right here. And the reason we look at this interaction in particular as well is that in comparison to measuring antibodies or antigens that are rather short-lived responses, if you look at the overall time of exposure, for example, to SARS-CoV-2, you will see that these responses are quite short-lived, and you have to be able to measure them within the next couple of days post-infection in your patient samples, which is not always doable. whereas a T cell response might last over several months. So this is something that you can look at long-term, and it is long-term immunity that we're interested in looking at here. So how do we monitor these cells? We use MHC multimer assays. So we will measure the interaction again between the TCR and the MHC peptide complex, either for class 1 or class 2, as we mentioned. In vivo, these interactions are very short. They have a half-life of about 10 seconds. But of course, in vivo, you would also have several complexes binding on the T cell at the same time, so which increases the time of interaction. And that's exactly what we try to do with these reagents, to simply mimic the antigen-presenting cell in order to be able to detect the corresponding T cell that will recognize these specific MHC peptide complexes. And over time, obviously, a lot of people, a lot of researchers have developed tools to study these cells, starting with dimers, tetramers, and pentamers. And what we're presenting today, the Dextramer technology, is sort of an advancement of that. So the first thing that you will notice is that we increase the number of peptide MHC complexes on the reagent. And that is to exactly increase the time of interaction with the T cell. And this is especially crucial for looking at low affinity binding T cells. In addition, we have a dextran backbone, so a sugar polymer that allows us to have multiple fluorophores on the reagent. So not just a single one, which obviously increases also the resolution and the intensity of the signal. Something we'll talk about a bit later is a second sort of advancement of this technology is to add DNA barcodes to the multimer to be able to simultaneously look at next generation sequencing and enhance sort of the multiplexing abilities of this reagent within a given sample. One more thing that I would like to mention is that different disease types will typically generate different affinities of responses based on the origin of the antigen. So we know that viral responses, for example, will generate high affinity T cells, and that's because we want the T cells to be able to recognize these pathogens very aggressively. So this is a natural sort of byproduct of how these diseases are developed. In cancer, you will see that the affinity for antigen-specific T cells is lower, simply because these are antigens as self-antigens. So obviously, we don't want a very aggressive immune response, but rather something a bit milder. So naturally, cancer epitopes or cancer-specific, antigen-specific T cells will usually have a lower affinity. And the same goes for autoimmune diseases. So what's important to notice is that depending on what type of T cell you're looking at, you will be more or less satisfied with the tools that you have available. We've developed the Dextramer reagents really to be able to cover this whole range of T cells, especially also those that are very tricky to enumerate and very rare in the blood samples that you look at. So here we come to really the core of the talk, which is our MHC Dextramer technology. So as I mentioned before, this is based on a dextran backbone, so a sugar polymer that simply allows us to attach an optimized number of MHC peptide complexes, as well as a label, a fluorescent label, to be able to use this reagent by flow cytometry. So we have this available in PE, APC, and FITC. And the complex itself is something that you will be able to customize based on the MHC allele that you need and the peptide sequence that you are interested in. And so with this tool, we have a high avidity for the T cells, obviously because of the high number of complexes, but also the Dextramer backbone stabilizes all of this and allows really the reagents to attach to the T cell for a longer period of time, again, to be able to make a sensitive reagent. So how would you use this? Very simply, this is mostly designed for cell suspension. So you could start you could start your experiment with PBMCs. You could also disrupt tissue to isolate tumor infiltrating lymphocytes or a cell line. And then it's a simple incubation of 10 minutes at room temperature that will allow the Dextramer to bind to TCRs on the antigen-specific T cells. So we'll look at some examples. So here, as I mentioned before, if one looks at high affinity T cells and sort of looks at CD8 positive T cells versus an MHC multimer, you can see that this population right here, that's the antigen specific population. In here, I think it was a CMV Dextramer probably. So you can see that both tools, Tetramer and Dextramer, can identify these cells. Dextramer with a bit of a higher resolution, so you can see this shift here compared to the positive population here. But when we look at the low affinity interaction, you'll see that the tetramer completely misses the cells. And that's, again, because the number of peptide MHC complexes is just not high enough to allow the cells and the reagent to bind to each other. So this is why we really recommend using Dextramer reagents for those low affinity interactions in particular. So let's look at an example that is a bit of a trend at the moment. So everyone talks about CAR T cells, and one of the key elements of CAR T cells is obviously that you express a TCR on the surface of a T cell. So you engineer it to express a very specific TCRs that is a target for a disease. And one way to make sure that your CAR T cell population is actually a pure population, one way to QC this cell line is to use Dextramer reagents to be able to quantify these cells. So in this paper, they were designing Treg cells, so CAR Treg cells that were also co-expressing IL-10. And these were HLA-A2 specific CAR Tregs. So this is just to show you the constructs that were used. And they wanted to express in particular a TCR that recognizes this combination here. And so with this peptide MHC complexes on the Dextramer backbone, so the Dextramer, we were able to help them measure the CAR Tregs. So you can see here, these were the IL-10 expressing CAR Tregs without our IL-10 expression, and then this was without the CAR. And you can measure the percentage of Dextramer positive cells, and you can see that in both the cases of the CAR T cells here, they measured about 90% of cells that were Dextramer positive. So which shows that indeed these cells are expressing the construct correctly and are exposing the TCR on the surface. So another example, more also of a trend here with COVID, lots of people have made these studies because all of a sudden we had these samples available. In this case, we looked at spike and at SARS-CoV-2 specific spike, right? And designed a panel of reagents. So not only targeted one epitope, but several different epitopes. So each reagent will have a single specificity, but you can use these reagents, same as antibody, you can pool them to make a pool staining, basically. And so from blood samples, we stained the cells with the pool of reagents and then looked at PE positive cells. So we have two panels here. One that we call non-spike, so that's all the epitopes that are not pertaining to the spike protein, but rather to the nucleocapsid, the membrane of the virus, ORF1, ORF6, and then we have a spike panel down here. So as you know, spike is used to immunize humans, right? So spike is the antigens that is used in most of the vaccines against COVID-19. So you would expect a spike positive response upon vaccination. And here we're looking at, let's say, a pre-pandemic sample where none of the panels can detect a response, right? So very low responses of T cells, which is normal. You would expect not to have any response. After SARS-CoV-2 infections, this is a post-infection for this sample. You can see that we have quite a strong non-spike specific response, which again is normal because when you get infected with all sorts of epitopes, not just spike, but also so you react to the nucleocapsid and the membrane as well. So quite a strong response here. And then after first and second vaccination, you can see here the spike specific response appearing. So what is interesting is that, and what it tells you is that in the end, maybe, getting infected, it protects you more or gives you a more strong response of a T cell response. But in the end, that really the vaccine as well generates a T cell response, a T cell specific response. So I don't know how I'm doing with time, but I'm hoping I have some few minutes to talk about the follow-up of these products. So we talked mostly about MHC Dextramer reagents. So these are really flow cytometry based assays. Sorry about that. And now we are looking at reagents that are compatible with next generation sequencing applications. So very briefly, we have on the same principle of the reagents that we've shown you, decode Dextramer reagents that are simply labeled with barcode. And depending on the barcode design, so the barcode format, we can look at large-scale screenings, so epitope screenings, for example, using a bulk sequencing approach. But we also have two barcode formats that are compatible both with the 10x Chromium and the BD Rhapsody, so single cell approaches. And those two will allow you really to be able to look at antigen-specific T cells and at the same time profile these cells via gene expression profiling, also have the TCR sequencing of these same cells and sort of do all this information simultaneously. So that's really the beauty of these single cell platforms, is to be able to get a lot of information on a few cells and have that all in the same workflow and in the same sample. So just briefly to show you again how the workflow works. So similarly as the COVID study I showed you, you would use different specificities, so a library of reagents to stain your cells. After staining, you would be able to sort or FACS sort or enrich your cells by selecting PE positive cells, because all of these reagents are still labeled with a fluorophore. And then once you sort the cells, you can decide either to do a bulk sequencing approach, so PCR and deep sequencing, to identify which of these specificities is enriched in your sample, or going a bit more deeply to one of the single cell platforms and generate libraries for each of these informations that you would like to have at the same time as the antigen specificity here. Another technology that we have that is a more of a do-it-yourself Dextramer. This is called dual-load Dextramer. So again, same principle as before. It's only that instead of providing you with a ready-to-use reagent, you can actually make the monomers yourself and assemble the monomers yourself in the lab. So the advantage of this is that you can work with smaller test sizes, that you can test different peptides on the monomers and see which one binds best. Maybe you're screening for different peptides that are similar but have different mutations, so you could do that also with your load. So this is very easy to use because you just load your peptide onto the monomer and then attach it to the backbone. And you can use the peptides that you have in the lab already. And one last thing we have is Dextramer Technology. So this is for those of you that want to go beyond T cells, beyond CD4, CD8 T cells, and look at more other types of immune cells or cancer cells. So this is sort of a very flexible tool based on the same Dextramer backbone, but you will notice that there's no peptide MHC complexes here attached. So it's an empty backbone with biotin acceptor sites that is ready to accept any monobiotin-related ligand. So this is to create your own multimer. And this is particularly interesting for B cells antigens. So we know of many customers that have loaded B cell antigens to detect antigen-specific B cells. But you know, the sky is the roof, as they say. You can load really any type of molecule as long as you have a target cell and sort of a fishing rod, I would say, to detect these cells. Then you can build a reagent that will work well for that. So one application of this obviously is antibody discovery, and this is exactly what was used here. So I'm just going to put this. So same workflow as before. We have a reagent that is barcoded, and for each new ligand that you put, you would have a unique barcode. In here, they only used one to label cells, sorted PE positive cells, and then did a single cell analysis using the 10x chromium to have simultaneously also gene expression profiling and BCR clonotypes. So this allowed, so this is data provided by Symphogen, a company in Denmark, allowed them to discover 21 new antibodies specific for the protein X that they loaded on the reagent. So this is again one of the applications of Dextramer, but as I said, this is a very flexible tool and can be used with any type of assay really. So with this, I will thank you for your attention. So I'm the technical application manager at Immudex, and I'm responsible also for customer support. So if you want to discuss your project with us, we're very happy to help, also jointly with Almog, obviously, and also brainstorming on the best experimental design. You can rely on our expertise for MHC peptide complexes. We've been doing this for 10 years now, and we have quite an extensive range of expertise in protein engineering. And of course, you can also reach out to Andrea directly. And please visit our website. It has a lot of resources, publications, but also case studies that will give you ideas of what type of application can be used for your research. So thank you so much, and I'll take any questions now. Oh, I can see the chat, sorry. Just gonna stop sharing and give the word back to you, Ajit.
In the chat, there is no question, just a big thank you from Gali. So I will join this thank you, and thank you very much for this great webinar today, Valerie. It was great having you with us. And thank you so much. Until the next time, I guess.
You're welcome. Thank you.
Thank you, everybody, for joining. Bye-bye.